for tuning in to this webinar on proteomics applications. For those of you who do not know me, I'm Paola Finch, the VGN Professional Development and Education Coordinator, and I help to organize this webinar in response to feedback from our funded faculty, where people ask for more research-focused outreach from VGN. With that being said, if you have any other ideas for webinar topics, please feel free to share those with me. And now I will introduce our Proteomics Core Director, Dr. Y. Lam, and let him give you his presentation.
Hey, hello everyone. My name is Y. Lam from the VGN Proteomics Facility at UVM. It's great to have this opportunity to share with you some of the work we have been doing at our facility. I might have worked with you in the past, but if not yet, I hope we can work together soon in the near future. As most of you know, our facility is supported by Vermont Genetic Networks, VGN, and the goal of our facility is to help investigators with their proteomics research, publishing papers, and to obtain funding. We also work closely with our professional development core and our bioinformatics facilities. The goal of our proteomics is to help investigators solve their research question using state-of-the-art proteomics technique. We have established an array of mass spectrometry-based approaches at our facility that are readily available to you and the community. Over the next several slides, I would like to briefly go through just a few things of what we do in our facility. This includes, but not limited to, protein characterization. If you have a protein, you want to know what it is or its characteristics, we can certainly help you. Analyzing post-translational modifications. Whether your protein is post-translationally modified, let's say acetylated or phosphorylated, for example. Looking at protein complexes, finding new protein-protein interacting partners. Large-scale quantitative proteomics, we can quantify proteins accurately using stable isotopes, looking at changes at the whole global proteome level. I hope by the end of this presentation, we will be able to get some insights into how proteomics or mass spectrometry-based methodologies can help your research. Again, we do protein identification. If you have a protein, you really want to know its identity, we can help you. The unknown can be from a regular one-dimensional gel or a two-dimensional gel electrophoresis where you can separate your proteins in a two-dimensional space, in this case, isoelectric form and molecular weight. And you find your proteins, target protein, to be differentially expressed between multiple conditions by gel-to-gel comparison. And you want to know what that protein is. We can help you to identify it. And also, we have worked with many investigators who want to confirm whether they're expressing the correct protein from a bacterial system or a mammalian system. The protein you may want to identify can be those that are immunoprecipitated down in an IP experiment, or in a pull-down experiment if your protein is epitope tagged. We can also identify proteins in an unknown mixtures. With the latest mass spec technology available, we can help you identify your proteins even you only have very small amounts of materials, let's say 1 to 10 femtomole, because these tiny little samples can get highly concentrated and then analyzed by our nanoscale liquid chromatography mass spectrometry LC-MS system, which is the instrument we use to make protein ID. By going through a so-called bottom-up approach, analyzing the enzymatically key peptides by mass spec, in the end, sequence information can be obtained, which allows us to come up with protein identification. I would like to point out that this kind of mass spec sequencing of a whole peptide takes less than a second nowadays, whereas 20 years ago, it would take an hour just to read one amino acid off a protein by Edmond degradation. So experiments that took months to accomplish 15 to 20 years ago can now be done within a day or two. As an example, we have helped one of our BPI investigators, Christina and Alan Trent at St. Michael College, to sequence and confirm her protein in her protein expression system. You can see in this slide the N-terminal peptide and the N-terminal portion of the protein was sequenced by mass spec. If you look at the mass spectrum, the spacing between the mass spectral peaks corresponds to the residual amino acid masses. So we confirmed her protein and we are all happy, and this work was published early this year. However, for most of the time, we would like to know more about our proteins, not just identify what it is. In fact, proteins can be pretty dynamic at the cellular level. As you can see in this slide, proteins can express differently across various cell types. They can be regulated at the post-translational level as well. Molecules can be phosphorylated in a signaling cascade. In this case, a kinase can be activated to phosphorylate a second kinase, and this second kinase can then go on to phosphorylate another kinase, which can ultimately phosphorylate and activate the target molecules. And the target protein can then perform its functions by either going into the chromatin to act as a transcription factor or binding to other proteins for signal regulation. On the other hand, proteins can be ubiquitinated and targeted for degradation. Secretory proteins can be glycosylated. Histone modifications like methylation or acetylation can regulate gene transcription. Not only that, under pathophysiological conditions like oxidative stress, free radicals like reactive oxygen and nitrogen species can be generated, which can modify proteins as specific residues. Change in protein expression or in post-translational modifications could regulate cellular mechanisms. Recent advances in mass spec-based proteomics can allow us to look at protein expression and post-translational modification globally and quantitatively in a way that was simply not possible 20 years ago. Elucidating how protein networks operate can help us understand many cellular disease mechanisms. So the goal of proteomics is to obtain a hopefully more complete and more integrated view of the dynamics of the proteome. So if you would like to study protein interaction, say you are interested in studying a protein, your protein, which is called protein A, you might want to study its protein interacting partners, what protein it is binding to. Since protein usually works with others inside a network of sometimes more than a couple hundreds of proteins. As shown here, protein A can bind to protein B in one complex, but it can also bind to protein C and in another complex. So by elucidating this protein interaction or connectivities, we might be able to understand how signal is transduced from outside of the cell to inside or from inside out, from extracellular matrix to intracellular to cytosol to nucleus and to chromatin. In this particular case, thousands of interactions have been documented. We can also study how protein interactions change when the proteins are stimulated or post-translationally modified, as shown here below in this schematics. If you would like to study protein interactions, we can help. We have worked with many investigators in the past who probably spent their whole career just working on one single protein, which might be very important, let's say in disease development. They would want to look at the differences in the protein, in their protein's interaction network, or differences in post-translational modifications between normal and disease states, or in wild type or mutant cells, for example. What they would do is that they would isolate their target of protein, their target protein of interest with an antibody, and run the immunoprecipitate on a gel and look for the differences by mass spectrometry. We can identify those interacting partners that come down with the target proteins in the immunoprecipitate that are present in not present in the control. For example, in this case, these are the IgG that used to immunoprecipitate your target protein, and these are the proteins that interact with your target proteins. Here's an example of using this strategy to find new protein interacting partners. An investigator from VGN Biochemistry Department was interested in the function of a protein called CBF-beta and wanted to look at its interacting partners to get some insight into what the protein does. So they ran the immunoprecipitate on a gel, and you can see the proteins that present in the immunoprecipitate, but not in the control. We help them identify these proteins indicated in red here. All they needed to do was to cut out the band and do the digestion, and it turned out that these proteins belong to several interesting categories. And they further investigated these interactions by co-immunoprecipitation and immunofluorescence microscopy. This is an interesting study, so we also published this like a couple of years back. On top of this, like I mentioned earlier, proteins can be phosphorylated, but it can also be modified by many other types of post-translational modifications. Different patterns of the same post-translational modification or different combinations of different types of post-translational modification on a single molecule, on a protein molecules, can recruit different sets of interacting partners, and these complexes can carry out various different functions accordingly. In this case, methylation and acetylation are important in gene regulation. Like some of you are interested in epigenetics, and ubiquitinated proteins can be targeted for degradation. If you want to know more about your protein and wanted to characterize its PTM post-translational modification, we can help you. And these PTMs can normally be characterized by their characteristic specific mass shift or diagnostic ions present in the mass spectrum. We have characterized many PTMs in our facility. Here are just a few examples. For example, like phosphorylation, acetylation, ubiquitination, phosphoserine versus serine. In the mass spectrum, phosphoserine can be seen as a mass shift on an extra active dalton added to the serine. There are a few other interesting modifications that we routinely characterize. For example, lysine residues can be methylated, acetylated, or trimethylated. And as you can see in this slide, interestingly, acetylation and trimethylation put very close masses on lysine residues. And you can see the difference is only less than 0.05 dalton, so it's very, very small. But however, we can use our high resolution and high mass accuracy instrument to distinguish these two. So it's pretty amazing what the new technology can do for us. Here are just a couple of mass spectra where we can clearly see where the modification is located. For example, phosphoserine versus serine alone. In this particular mass spectrum, you can see that the lysine is acetylated with an addition of 42 dalton to the lysine residues shown here. Not only do we identify proteins, we also do quantification. So we can quantify hundreds to a few thousands of proteins in a single experiment with stable isotope-based quantitative proteomics. And if you would like to know what the protein differences are between two sample populations, yes, we can help you. We can label two or multiple proteomes you would like to compare with a stable isotope mass tag that comes with two or more versions, one heavier than the other one. In this case, light label for proteome A and heavy label for proteome B. As these two proteomes are labeled differentially, the same peptides from the same protein, but from two different samples, two or more different samples can be distinguished by their mass differences using mass spectrometry as shown here. In this particular case, this protein is more abundant in the heavy sample, whereas this protein is equally abundant in both. The advantage of this technique is that hundreds to thousands of proteins can be identified and the expression accurately quantified in one single experiment. We can further interrogate the differences using software support like commercially available software and of course expertise from our bioinformatics facility. Also, this labeling method can be applied to cell culture study, not only cell culture study, but also to animal models, human specimens to study protein expression, and as well as stoichiometry of protein interaction and PTM globally. So there are multiple ways to introduce labels into the proteomes, either by taking advantage of the endogenous cellular metabolism or by chemical labeling. I won't go into details for now, but briefly, if you're working with cell culture models where you can culture your cells in an amino acid deficient medium, typically lysine and arginine deficient medium, because we use trypsin. And then we can put back the light unlabeled and heavy stable isotope labeled amino acid into the medium. And this isotope labeled lysine and arginine would then be incorporated through endogenous metabolism into the proteome after 5 to 6 doublings. By such differential isotope labeling, proteome changes under different conditions can then be determined as shown here. Right? This particular protein is more expressed in the treatment as compared to the control. The isotope can be incorporated into the proteome chemically as well. I would like to share with you today one of the examples using this stable isotope strategy. This is a collaboration our facility has with one of the investigators in the microbiology department. We are looking at the differences in the proteins of our non-pathogenic bacterial strain versus its pathogenic counterpart. The pathogenic strain has one membrane protein called MOR-C, which is mutated and causes disease. So we would like to look at the differences in the membrane proteome and hopefully the secretory protein profile between these two bacterial strains. So the workflow goes like this. We extract the protein from wild type and mutant cells and label them with light and heavy tags, like I mentioned to you earlier, and then apply the workflow of quantitative proteomics that are described in the last slide. And to keep the story short, we identify and quantify 700 proteins, with 12 of them differentially expressed between wild type and mutant. In this heat map, each row represents one protein. Proteins that are upregulated in mutant are highlighted in red, whereas downregulated ones are highlighted in blue. The downstream effector leukotoxin of the mutant gene MORC was diminished in expression. So our investigators further validate some of these expression changes found by the mass spectrometry by Western blot analysis. And they were very excited about the findings because this data actually provided them with hypotheses for future experiments, as well as data for grant applications. And we published these findings a couple of years ago in the Journal of Proteomics. We can use this stable isotope labeling strategy to look at the differences in protein interaction networks or the interactons as well, and quantify the proteins that are binding to wild type and mutant in this case. In this cartoon, you can see that the wild type binds 3 proteins, right? But when an amino acid is mutated, it only binds to 2 proteins. So the differences between wild type and mutant can be quantified using proteomics. I want to show you one of the examples. This is a collaboration again with the VGN Biochemistry department. The investigators isolate their target proteins of interest by antibody and wanted to look at the changes in the interacting partners between wild type and mutant proteins. Because the target proteins are active targets, so anti-FAG antibodies was used for the immunoprecipitation. We ran the immunoprecipitate from wild type, mutant, and control onto a gel and help identify those proteins that came down in the IP by mass spectrometry. In this case, we identified 150 proteins that bind with different amounts to wild type and mutant. About 100 of them bind more to wild type than mutant. So this is a pretty interesting study. And I wanted to know that my colleagues and at the facility and myself have many years of experience in proteomics and have established a number of workflows on analyzing protein expression, post-translational modification, as well as large-scale quantitative proteomics, and we will be more than happy to work with you on all these aspects. Thanks to VGN, we were able to keep our instruments state of the art for the past 10 years, and advanced mass spectrometry instrumentation Q-Exactive was installed almost 2 years ago, and we will be installing another one next month pretty soon, and that's pretty exciting. Such new instrumentation can allow us to compare quantitatively or to multiplex 10 samples in one experiment. We can even compare more than 10 samples in multiple experiments. So it's a really powerful technique. For now, I won't go into the chemistry or the methodology, but this is some data we obtained from a recent collaboration we had, where we are looking at a particular drug effect on the expression of a couple hundred proteins in a cell-based system. This type of analysis allows us to have a more comprehensive view at the mechanism of that drug effect at the protein level. So please don't hesitate to let us know if you think we can be of any help with your research projects. We will try our best to assist you in any way we can. If you're ever in the region or in Burlington, feel free to stop by our facility anytime. We are located at Mars Life Science Building. So our doors are always open, and I would like to emphasize that we are the only facility in the state of Vermont equipped with such a high-end instrumentation. Just want to show you a little bit of what we can do for you. We can perform accurate mass measurements. In this case, we were able to tell like the elemental composition of a particular compound, how many carbon, hydrogen, and oxygen, with a precision down to the third decimal place, like 0.0005 Dalton. To give you an idea, is the precision is the mass, the precision we can detect is close to the mass of an electron. So we can access to a total worth of more than $2 million of instrumentation, and that we only charge $10 for processing one sample to regular user, and we do it for free for our VGN partners. I think we should all make good use of our facility, and you can check out our website at vgn.uvm.edu, and below is our contact information. This is my colleague, this is myself, this is me, this is my colleague Dean, and this is Katrina. And we are all very happy to help you with your research projects. And some of you might have worked with Dean before in the past. I would like to use this slide to show you that we not only provide expertise to investigators, but also for other investigators, mostly in the New England region, including Dartmouth, Jefferson Laboratory, and UMass Medical School. You see, we also have one collaborator from Argentina as well. I think we have built up a nice reputation since the inception of our facility in 2006. We have analyzed over 20,000 samples, around 2,000 samples per year, and we have interacted with more than 80 investigators over the past few years, and we have helped investigators to publish more than 100 papers in total, and with more than 30 papers over just the last 2 years. Currently, we are working with a number of investigators from BPI. I hope we can work with you in the near future. And we provide consultation. We not only provide consultation, we also provide expertise for experimental design, data analysis and interpretation, all the way to manuscript preparation. Again, we also work closely with the professional development core in our Proteomic facility to do a lot of outreach activities at many of our Vermont local colleges to educate our undergraduate students, our future scientists about proteomics. And as you can see, we have provided more than 70 support letters and are supporting more than 20 active grants from various funding agencies. So here's a slide showing a quick snapshot of the researchers we have worked with over the years. I didn't have enough space to put all the researchers we have worked with on just one PowerPoint slide, but these are the ones that we have featured in our VGN newsletters. It looks like on the photo that they were all smiling and we were all happy with they were all very happy with our expertise. As most of you know, our facility is supported by Vermont Genetic Network, and our principal investigator is Dr. Judy Van Hilton. Again, feel free to contact us. I will be more happy to take questions, learn more about your research, and to help. Thank you very much.
Okay, so we have a question from Bob Cluck. Bob asks, are you able to determine the native molecular mass of a protein? Monomer, dimer, tetramer, etc.
So it depends on the molecular mass of the protein. So it's a small protein, we can do that. But it's a bigger than that, it will be a little bit challenging. But it depends on what type of experiment you want to perform, I think. So how big is the protein?
So Bob, we go ahead for asking a general question, or you have one specific protein in mind that you want to?
Okay. So the monomeric molecular mass is anywhere between 38 and 50 kilodaltons.
Okay. So you won't be able to look at the monomer, dimer, or trimer using like native gel electrophoresis or.
Yeah, we've done that, but I mean, we can do this by NMR, I guess, but you don't have
Oh, yeah, you're right. That's the.
Okay, but
Yeah.
there isn't a way, given the instrumentation and methodology in the lab, that you can do it there.
You're right. So that's a good question. So currently, most likely we won't be able to do this type of analysis. Probably the most straightforward of doing this type of analysis is use a multi-top. So basically, like those multi-top, they have unlimited mass range.
Okay.
So like a instrumentation where they can like usually mostly used for intact mass analysis.
Okay.
I don't know whether it is.
So there isn't an opportunity. I'd have to go elsewhere to have that done.
Yes, but do you really need to get accurate molecular mass or?
Well, I'd like to. I mean, we're doing gel filtration right now, but
You're right, exactly. So I was about to say, like, people usually do gel filtration. If you know that, like, if you run it from the gel filtration, if you know that it's a dimer, then you basically just run it to on a SDS page. If it on a reducing condition, if it exceeds a monomer, then it's a dimer. So usually people do it that way. If you don't want an accurate molecular mass determination.
Yeah, I can cross-link too and look at it that way. I just was wondering if there was a way to do it directly with an instrument. So since I'm wired for sound here, let me ask another question. How sensitive are you able to detect post-translational modifications? Let's say the percentage of modification at a particular amino acid residue is only 10%. Can you pull that out from all of the unmodified residues?
You're right. So it depends. So usually, like for example, phosphorylation, like the serine or furan phosphorylation probably is about like 20 to 30%, but tyrosine phosphorylation can be as low as 1% to 5%. So it depending on how you want to proceed the experiment. Some people will use like a motif, a specific antibody to pull down the phosphorylation or acetylation, and you want to look at and just try to like identify as many as possible.
Mm-hmm. Okay, thank you.
Thank you, Bob. So looks like we have a question from Norwich University. I'm going to unmute the microphone for Norwich. I'll ask the question, and then if you have follow-up questions or want to interact, please use your microphone. The question is, does it have to be a mammalian protein?
No. So any protein, as long as the genome is completely sequenced, then we should be able to identify them. So I didn't go into detail on how this mass spectrometry based like identification work. So what we usually do is that we do a triplet digestion where we can sequence all the peptides from the particular protein you are interested in, and then we can like search the mass spectrum against a database. So if the genome of your organism is completely sequenced, then we can basically match the experimental mass spectrum to the theoretical mass spectrum generated from the database. So in short, is that any organism with a complete sequence, with the genome complete sequence, we should be able to identify the protein complements.
Okay, we just had someone ask to repeat the question. So why does David answer to the question, does it have to be a mammalian protein? Norwich University, I don't know if you have any follow-up that you want to ask from that. Your microphone is turned on.
So can you hear us?
Yes.
Okay. So it's just to follow up, this is Karen. Hi, Y.
Hi, how are you?
Good. So one of the things that I think is so unique about this is the kind of unique and creative projects that utilize proteomics. And the one that I was thinking of is the WASP and pitcher plant project that Amanda is working on.
Yeah.
So could you just give a really brief overview of that one? So she goes out into the field and collects specimens, right, in the field, and then what is the whole premise of that project?
I'm not particularly involved in that
Okay.
particular project, but from what my understanding is that they want to look at that particular they want to look at the particular protein that are present between different plant species, and
Okay. I mean, that's what I wanted faculty here to think beyond the biomedical aspect of it, because it's such a powerful method to look at even field species and things like that. So.
oh, you're right. So basically, the idea is that so any experiment that involves in protein analysis, and then you have if the particular organism that have a genome that the genome is completely sequenced, you pretty much can use proteomics methods to interrogate the relationship of the protein relationship in that particular experimental settings.
Right. Okay. I have
So
one more.
most likely, like I think if the genome is not completely sequenced, we still have other ways to go around it properly by searching the mass spectrum against a database that are of a species that are closely related to the species you are working on.
Okay. And then one other question, and that's how much of a role do you play in experimental design before? Because I know with the microarray facility, they used to really want to be involved in experimental design to ensure that on the back end that everything was in line for the.
You're right. So most likely, like because from our experience, most of the investigators, if they want to do proteomics, before they want to even think about the project, they will come here to talk with us first. So we always have a consultation section with the investigators and talk about the experimental design and how long does it take to get the result they expect. I mean, what kind of results they can expect and what kind of result we can actually deliver to them, and how long does it take, and what kind of result can be actually published can be published, and what kind of resources they need to provide in order to make the experiment successful.
Okay, thank you.
Thank you. Do we have any other questions? Okay, it seems like most of the participants have actually asked a question. So with that, we hope that this is just the beginning of the dialogue, and we really encourage you to reach out to I and the Proteomics course to continue this discussion on your own research project. And so we thank everyone for attending, and we hope to see all of you that intend to apply for a project or pilot board in February at the grant writing workshop that is on November 11th. Again, we will post this recording up on our website, and I will send out an email. And we encourage researchers that are considering to submit an application to us next year to watch this webinar and think about how they might be able to include this free service into their applications for next year. So thank you, Y, and